All right, macromolecules, and this time we're looking at nucleic acids. So nucleic acids are made up of the monomers that make them up are called nucleotides. The individual units are nucleotides, and when you join those nucleotides together, the bond that is created between them is called a phosphodiester bond, and this forms this backbone. We refer to the backbone itself as a sugar phosphate backbone, and we'll see why when we look at what the nucleotide is made up of. We're looking at two types of nucleotides that you will recognize, hopefully at least one of them, DNA being one of them, and the DNA stands for deoxyribonucleic acid. And we're also going to look at RNA, which is not deoxy. It does have the oxy, and so we just call it ribonucleic acid. Often the DNA contains the genetic information of the organism, and we can talk about viruses in more detail, but in terms of how they even count as an organism or not, but they may use RNA. But for things like humans and other eukaryotes, we're going to have the genetic information encoded for in the DNA, and then the RNA, which is the other type of nucleic acid we're going to be looking at, it will be used to take that information from the DNA in order to make proteins. So the monomer itself, this is a sort of simplified version of it, but there are three parts that we need to be aware of. There's the phosphate group on it. There is the 5-carbon sugar, hence the phosphate sugar backbone. And then importantly, there's also a nitrogenous base added to it. And this is where the variation is going to come within the nucleotides of, say, DNA or of RNA. The sugar itself, again, we could have the deoxyribose version, that's what DNA uses, and then there's the ribose version that RNA uses. And you'll notice that the difference on them is that this carbon number 2 on DNA just has a hydrogen, whereas carbon number 2 on RNA has a hydroxyl group, and that's why the deoxyribo is the DNA version of it. The nitrogenous bases themselves come in 4 varieties. There's some odd ones and whatnot, but we're focusing on these 4 main ones that you make up the bulk of DNA and RNA. And so the nitrogenous base is the part that changes. It could be a thymine, it could be a cytosine, it could be an adenine, or it could be a guanine if it's DNA. For RNA, it uses some of the similar bases. So you see that cytosine, guanine, adenine are the same. And then what's different is that one of its nitrogenous bases is uracil. So RNA uses uracil, whereas DNA uses thymine, and the other 3 are the same. So these are the 4 nitrogenous bases that either DNA will use or RNA will use. The phosphates are going to be identical for the DNA and the RNA. So the bond that holds these nucleotides together to build this chain of nucleic acids that is your DNA is a phosphodiester bond, and it's going to be using the phosphate group of the nucleotide and the hydroxyl group of the sugar, and it's on carbon 3, because remember, DNA doesn't have a hydroxyl group on carbon number 2, so it wouldn't work with carbon number 2 for DNA. RNA does have a hydroxyl group on here, but it is actually using carbon number 3's hydroxyl group to do it. And again, this is a dehydration synthesis, so it's going to be putting those nucleotides together, so we have to remove a hydrogen, an oxygen, and another hydrogen. So imagine those 3 coming out of here. And then the oxygen on the phosphate is now going to be directly bonded to carbon number 3 of the sugar. Whether that sugar is ribose or deoxyribose, it would be the same thing. So this gives us our sugar phosphate backbone. So we have sugar phosphate, and it's connected to another sugar phosphate, connected to another sugar phosphate, connected to another sugar phosphate. So that is the sugar phosphate backbone of DNA, and that's what is going to start to cause this to spiral in on itself with these nitrogenous bases. Let's get a different color here. Nitrogenous bases sticking out from the sugar phosphate backbone. And remember, it is the nitrogenous base that determines which nucleotide you have, and that is where the information for our genes are encoded. The order in which those nucleic acids are positioned with their respective varying nitrogenous bases, that's what's going to carry the information. So DNA itself, when it forms, it's going to be double-stranded. So you have these two parallel strands of nucleic acid chains running along each other, and they are going to hydrogen bond to each other. So one strand, and I'm sure you have seen DNA depicted as a ladder. That vertical part of the ladder is a sugar phosphate backbone, and the rungs of the ladder, the part where you'd step on a ladder, are the nitrogenous bases. So one ladder, let's get a different color here, is going to line up, one side of the ladder is going to line up with the other side of the ladder that also has its nitrogenous bases forming, facing into the center. And it is hydrogen bonding that holds those two pieces together. The nitrogenous bases, here they are here, there's your hydrogen bond, they are going to hydrogen bond to each other, and that is what's going to hold the two halves of the ladder together. Adenine will hydrogen bond to thymine, and guanine will hydrogen bond to cytosine. So that is how they match up, one half of the ladder to the other half. This in DNA is going to spiral, so this eventually, when you get those hydrogen bonds formed, is going to form a spiral structure, and this is why we call it a double helix. It's helical, and there are two parts to it, so it's a double helix, the two sides of the ladder. RNA is going to be single-stranded, and so it does not have this complementary basing. It can have a secondary structure to it, where you get the chain and the order of the nucleotides, and then they can clump up, kind of like proteins do, and that gives RNA some ability that DNA doesn't have to actually function like a machine.